Hi, I will now talk about the anaerobic fermentation working group, which I co-lead with Luca. Our group is supported by both the Environmental Biotechnology Network and the Biomass Biorefinery Network. Thanks to their strong support, we have been able to run a range of activities to bring people together, to encourage collaboration, and to share knowledge and experience. First of all, I would like to explain why we are interested in anaerobic fermentation. Fermentation, as we all know, covers a wide range of bioprocesses, but our focus is quite specific. Mixed culture anaerobic fermentation of multiple putrescible organic waste and residues using robust microbial communities without pre-sterilization. We believe this biotechnology has potential to address a critical gap in industrial-scale bio-based organic waste management because it complements two other major types of biotechnologies, which are the precision bioprocesses for high-value products and anaerobic digestion for energy recovery. Volatile fatty acids and plant nutrients such as ammonia are the main products of this process, and it is also regarded as dark fermentation in some cases. Just to explain, the working group also focuses on process integration and resource efficiency, aiming to minimize the external chemical inputs, especially during the product recovery stage. Several sectors can benefit from the development of this anaerobic fermentation, we believe, for example, the anaerobic digestion sector, because it can help transform the sector from an energy-driven into a material-driven dirty biorefinery. Well, we can argue that the biomethane itself can be regarded as a platform chemical, which I totally agree. Actually, anaerobic digestion is one of my all-time favorites. But in future, transforming anaerobic digestion to fermentation may improve the profitability in some scenarios, and on top of that, it can also add flexibility in terms of our process choices, especially for challenging waste streams. For example, the high protein materials such as slaughterhouse waste or fish waste, or the high salt, high sulfate streams such as the seaweed or microalgae residues after the extraction of high value elements out of it. Of those materials are unlikely to be used as single substrates for the digestion process, but they are ideal for this fermentation. Fermentation and digestion are therefore complementary in this case. For all these reasons, we believe mixed culture anaerobic fermentation has real practical significance, and we want to promote it. Over the past 12 months, we have been quite active. Here are some of the activities we have conducted. We hosted an invitation-only workshop. We created an animation to explain this anaerobic fermentation in a simple, accessible way. We also carried out two small studies. One looked at how emerging biotechnologies affect material choice for bioreactors, and the other explored integrated chemical-free production and recovery of fatty acids and ammonia. We also organized a webinar to get some insight into several Horizon 2020 projects related to the innovation and deployment of this anaerobic fermentation and to build connections with researchers in other countries. I would like to talk a little bit more about the workshop. Its primary aim was to identify research gaps and opportunities for this anaerobic fermentation. We focused on 3 case studies, each based on a different feedstock, which are wastewater biosolids, municipal solid waste, especially the source-segregated food waste, and agriculture, industrial waste and residues. Some common themes came up from fundamental research to the process integration to market and policy development, which need to be addressed now and in the near future. It is also important to note that each feedstock has its own characteristics and management context. For example, fatty acids produced from wastewater biosolids can support the biological nutrient removal from wastewater, so play a useful role in wastewater treatment itself, and therefore would be good to get promoted. On the other hand, however, we also need to keep in mind some potential concerns and wider considerations, such as how to integrate this fermentation effectively into the existing process chain and how to deal with some other concerns, for example, emerging pollutants in this case. Overall, the workshop was really productive and opened the doors to new collaborations. Looking ahead, what's next? Both networks will wrap up early next year. The positive aspects is that our core members are committed to staying connected and looking for new funding opportunities. And in the meantime, there are things we can do as a working group without a budget, for example, webinars. So if you have presentation ideas or want to collaborate, we'd love to hear from you. Thank you.